Inheritance, Part 2. Genetics is the branch of biology that studies heredity. Heredity is determined by the genes that are passed from parent to daughter cells. Genes are pieces of DNA that code for the production of a polypeptide molecule. This code is embedded in the sequence of bases within the DNA. A triplet of bases stands for one amino acid in the protein that will be constructed on the ribosomes in the cell. One chromosome contains enough DNA to code for many polypeptides. If chromosomes contain DNA that make the same polypeptides, they are said to be homologous. This means that in a diploid cell, there are two copies of each gene. These copies of each gene lie in the same locus on both homologous chromosomes. So as you're looking at the diagram, you have homologous chromosomes. You can have a gene, okay, for this capital Y and lowercase y, they're both the same type of gene, but it doesn't mean they're going to be the same allele. Each species has a specific number of chromosomes. Human cells contain 46 chromosomes, 2 of each 23 types. Each type is assigned a specific number and contains its own particular genes. In order to have the total 46 chromosomes, a cell obtains 23 from the mother, which is the maternal chromosomes, and 23 from the father, the paternal chromosomes. There are different forms or varieties of genes, and these are known as the alleles. Again, looking at your diagram, you can have the same gene and the same locus, but it could be a different allele. So this gene is representing the color of the flower, but purple, and this gene is still representing the color of the flower, but it's white. So both the genes are for the color, but the allele, the specific color, is different. Looking at genotypes and phenotypes. Most genes have several different alleles. To make things simple, the different alleles of a gene can be represented by symbols. Looking at the HB raised to the A, capital A, or HB raised to the capital S, the capital A, this is indicating an allele for normal beta polypeptide. In other words, somebody without sickle cell anemia. If there's the HB that's raised to the S, then the allele is going to represent sickle cell polypeptide. The HB is going to stand for the locus on the hemoglobin gene. The superscripts stand for the particular allele of the gene. The alleles that an organism has compose its genotype. When two alleles of a gene are the same, they're said to be homozygous. When the two alleles of a gene are different, they're said to be heterozygous. So looking at our examples, these would represent homozygous, normal. The second example is representing heterozygous, still normal. And then this last one is homozygous, recessive, so it would actually represent somebody with sickle cell. The observable characteristics of an organism is the phenotype. The physical characteristics, think P and P, physical characteristics, the phenotype, this is what you actually can observe. So in an individual that's going to have the two uppercase A's, that's going to be somebody with normal blood cells. Somebody that has the capital A and the S, this is going to represent heterozygous. So we have an individual that's a carrier, but they're normal due to the dominant trait. And then if you represent both recessive traits, that's homozygous, recessive, this individual, this is going to represent a sickle cell anemia. Inheriting genes. Gametes during sexual reproduction are created from diploid body cells. Therefore, each gamete will carry one chromosome of each pair. This means that each gamete has only one copy of each gene. Depending on the genotype of the individual and the gametes that can be produced by the individual, the possible genotypes of the offspring can be predicted. So in this case, we're crossing two heterozygous. This is representing heterozygous. This is also heterozygous. If you were to complete the Punnett square, then you would see that this individual is going to be homozygous dominant, no sickle cell anemia. The second individual is a carrier, but again, they're not going to have sickle cell anemia. This one, again, is heterozygous, where the individual is going to be a carrier, but again, because of the dominant trait, no sickle cell anemia. And then for this last one, this one is homozygous recessive. This is going to represent an individual with sickle cell anemia. Again, please remember with Punnett squares, these are only probabilities. They're not guaranteed. We don't know exactly which gametes are going to be involved in the fertilization process. The standard way of showing the genotypes of offspring that might be expected from two parents is to look at a genetic diagram. Genetic diagrams will always include the parent phenotypes, that's physically the physical characteristics you can see, the parent genotypes, that's going to be the letters that are representing the alleles, the possible gametes for both parents, the possible offspring genotypes with the probabilities for each, the possible offspring phenotypes with the probabilities for each, and then a Punnett square is constructed to predict the above information for the offspring for the chosen parents. Looking at a Punnett square, you have your parental phenotypes, brown coat, brown coat. The genotypes, these are the allele letters, capital B, lowercase b, meaning brown is going to be dominant, capital B, lowercase b. So both of the parents are going to be heterozygous brown. The gametes, these would separate into gametes, so you could have capital B or lowercase b for both gametes on both the mom and the dad. Setting up your Punnett square, you put your gametes for the female on one side, the male on the other side, and then you complete your Punnett square, bringing them together. Two capital B's, homozygous dominant, brown coat. Then we could have, in this case, two of the possible offspring would be heterozygous brown coat, and then a fourth option would be homozygous recessive white coat. So our predicted offspring would be 3 brown coats to 1 white coat. Taking a look at dominance, there are instances where both of the alleles of an organism have an effect on the phenotype of the organism. This means that the alleles are said to be co-dominant. When dealing with this scenario, you write the genotype of the organism with a capital letter for the gene and then a superscript that's going to correspond with the allele. So this capital C is indicating that we're looking at the color of flowers. And then the superscript is going to tell us the specific allele. The capital R is representing red flowers. The capital W is representing white flowers. But since both of these traits are dominant, they're said to be codominant, meaning it is possible when you are crossing flowers that are red and with white, that it is possible to get one that's going to be codominant, where the red and white are going to actually give us a pink flower. There are instances where only one of the alleles has an effect on the phenotype of the organism. This means that the allele is dominant. If the allele has no effect on the phenotype of the organism, the allele is said to be recessive. So if you're not looking at codominant traits, you're just looking at the dominance, this would represent heterozygous dominant, homozygous dominant, and then homozygous recessive. When dealing with this scenario, you can assign a capital letter for your dominant allele, lowercase letter for your recessive allele, and then make sure you differentiate between them. You need to make sure that you write them very clear, one uppercase and one lowercase when needed. Can't have them resemble one another. Test crosses. Unfortunately, when alleles show dominance, it's not always possible to be able to tell the genotype of the organism with a dominant allele just by looking at it. In order to determine the actual genotype of the organism, you need to do a test cross. A test cross involves crossing an organism, showing the dominant trait with an organism that is homozygous recessive. In other words, you're completing your Punnett squares. So when you're looking at a purple stem and a green stem, again, you could have the purple here is dominant, green is recessive. So in heterozygous, you would have a purple stem because of that dominant purple color. These are both capital A, so it's homozygous dominant purple. And in this case, you have two lowercase letters of A, therefore this is going to be homozygous recessive green stem. Most genes have more than two possible alleles. A gene in this situation is known to have multiple alleles. Blood groups are a perfect example of this scenario. Blood groups are actually composed of 3 alleles. 2 of the alleles, you have I, capital I with the superscript capital A, and then you can have the capital I with the superscript capital B. These are codominant. The other allele is the capital I with a superscript of the O. This is recessive. The I stands for isoagglutinogen. It's another term for an antigen that's going to code for an enzyme that modifies the carbohydrate content of the red blood cells. Looking at the genotypes, again, your genotypes are represented with the letters. Phenotypes is the physical characteristic. So here, when you're looking, you have the codominants, 2 capital A's or a capital A with the recessive O. Both of these are going to represent an individual with A-type blood. With the capital B's, having both capital B's or a capital B with the recessive O, both of those represent B-type blood for phenotype. You can have the codominant of AB, where this individual's blood would be AB, or you could have O-type blood from 2 recessives. Couple questions for you. If you would please pause the video and go ahead and complete these 2 questions before continuing. Looking at an F1 generation, F2 generation, and test crosses, your F1 generation is the offspring that result from a cross between individuals. It doesn't necessarily have to be a homozygous recessive and homozygous dominant genotype. It just means that there was a parental cross that gave us an F1 generation. The F2 generation is going to be the offspring that result from the F1 individuals having a cross. To put that into perspective, kind of to give you an idea to help you with that, think of yourself as being the F2 generation. The F1 generation would be your parents, and then your grandparents would be the parental generation. To kind of break that down, just to give you an idea what that is referring to. Test crosses, this is actually doing your Punnett squares, where you would take the gametes, cross them. This allows you to see if the offspring are going to be homozygous recessive, heterozygous, or homozygous dominant. Okay, a couple more questions for you to answer. If you'll go ahead and pause the video and answer those two. Sex inheritance and linkage. Sex chromosomes in a cell are not always alike because they do not always have the same genes in the same position. This is due to the fact that there are two types of sex chromosomes, the X and the Y chromosome. Remember, males are going to be XY, where females are going to be two X's. The X chromosome is larger and it carries more genes than a Y chromosome. The genes that are carried by the female sex chromosome are said to be sex-linked. So anytime you find a gene that's attached on an X or a Y chromosome, those are sex-linked genes. Sex-linked gene is one that is found on the X chromosome that is not going to have a match on the Y chromosome. When dealing with this situation, sex-linked genes are represented by symbols that show that they are found on the X chromosome. An example would be hemophilia. Hemophilia is usually represented, it's a recessive trait, it's represented usually by an H, also known as factor VIII. Lacking it would be the recessive. Dominant is without hemophilia, and it's sometimes also known as the normal factor VIII. All right, taking a look at hemophilia, I know I just said that it's usually represented with H, but this is a diagram out of your textbook where they're using an F. So we have our parent phenotypes, a normal female and a normal male. With the male, again, it's an X and a Y chromosome, and then there's the capital F, meaning normal, so this individual's blood will clot normally. For the female, you have two X chromosomes. This female has the capital F, so that's the dominant trait for normal clotting of blood, but the female is also a carrier because of this lowercase f. Does not mean the individual has hemophilia, just that they're a carrier. These are representing the genotypes. This is representing the phenotype. The gametes, literally when you're separating out your gametes, put the male on one side, female on the other side. For this first possibility, you have a female that's produced. In this case, this is going to be a female with normal blood clotting and not a carrier at all because they're carrying both capital F, so both of the traits for normal blood clotting. The second female that's produced, again, is still going to have normal blood clotting, but she is going to be a carrier for hemophilia. Taking a look at the males, for this first male, you have that capital F, so normal blood clotting for this male. There isn't anything on the Y because it's not a Y sex-linked trait. It's only associated with the X chromosome. This is a typo in your book. This diagram's from your book, so please note as we're going through this. This F should be lowercase because it's coming from this gamete right here. That's a lowercase f, so that should be a lowercase f right here. Since this male, XY is a male, and again, that should be lowercase f, has the lowercase f, that's going to represent a male with hemophilia. Hemophilia is the inability to have your blood clot. So, typically when somebody skins their knee, the blood starts to dry and a scab forms. Someone with hemophilia, their blood would not clot, that scab would not form. They would need to have medication to help them. So this male, again, that's a typo there, that should be a male that is hemophiliac, indicating by the recessive non-normal sex-linked trait there. So we have two females with normal blood clotting. Again, one does have a carrier though, and then one male that's normal, and then a male with hemophilia. Looking at the females, because they have two X chromosomes, you have three possibilities. Okay, this is a normal clotting individual. This one, also normal clotting because the dominant trait for normal blood clotting, but again, also a carrier. This individual, because they have both of the two lowercase f's, this individual would have hemophilia. However, in females, it is rare for a female to be born with hemophilia. It's possible, but again, it is rare. The second one, looking at the males, again, we have the normal blood clotting male, and then this male is going to represent the hemophilia. There you see that lowercase f. Again, that is a typo right there. Okay, a few more questions. If you would go ahead and pause the video and answer these questions before moving on. Dihybrid crosses. This involves the inheritance of two genes at once. When cells undergo meiosis to produce gametes, the pairs of homologous chromosomes line up independently of each other in metaphase 1. In many cells undergoing this division, it is likely that the chromosomes in roughly half of the cells will line up one way and the other half will line up in another way. This is your independent assortment that occurs during meiosis. This way you can predict what the gametes are going to possibly be with approximate equal numbers. So again, with dihybrids, you're looking at two different genes at once. Here we're looking at purple stems or a green stem, and then cut leaves, meaning a jagged edge, or potato leaves, meaning a smooth edge. So if we were to cross two heterozygous parents, okay, heterozygous for both traits, both of the genes, we would have a purple stem and then a jagged leaf. Using that same example, the heterozygous parents, okay, both are indicated still. To determine the gametes, you use FOIL. To get FOIL, you would go your first, remember your first is going to be this capital A, capital D, the first in each of those alleles. So there's our capital A, capital D. Then you would use the outside. So our outside is going to be capital A. The outside over here is lowercase d. So there's my capital A, lowercase d. Inside, lowercase a, capital D. Lowercase a, capital D. And then last is going to be the last one in each of them, the lowercase a, lowercase d. Lowercase a, lowercase d. In this case, since both parents are heterozygous, their gametes are going to be the same. Using this FOIL method is going to allow you to get all possibilities for the gametes. If you had one parent that was heterozygous and then different set of circumstances for the other parent, you would have to FOIL again. But in this case, since they're both heterozygous, you're going to make your Punnett square. In dihybrids, you're going to have a total of 16 boxes. So you put your gametes for one parent across the top, gametes for the second parent along the side, and complete your Punnett square. Keep the same letters together. Don't do this mix like AD So here you wouldn't go capital A, capital D, capital A, capital D. That will get very confusing. Okay, keep the same alleles together so it's easier for you to look at it. Typically when you cross two heterozygous parents in a dihybrid, you're typically going to get this ratio 9:3 to 3:1, meaning in this case you're going to have 9 purple cut leaves, 3 purple potato leaves, 3 green cut leaves, 1 green potato leaf. So this 9:3 to 3:1 ratio is typically, again, what you would see from a heterozygous, both parents heterozygous. All right, some more questions for you guys. If you would pause the video and take a moment to answer those questions. Epistasis is the interaction of two genes at different loci. One gene may affect the expression of the other. An example, the colors of feathers in chickens. There is an interaction between two gene loci on different chromosomes. So we have two different chromosomes where we're representing the gene where we have a capital F, lowercase f, and capital G, lowercase g. The capital G is going to determine the feathers have colors. The recessive G, lowercase g, means there's no color, they're going to be white feathers. And then the capital F is the dominant that's going to prevent the production of color in feathers. Even if there's a dominant color showing, indicating that it has the allele, this capital G, and we should see a colored feather, if it has this capital F, the colors can't be produced, therefore we're not going to have colors. So when you look at the possible genotypes and phenotypes, these first several genotypes, typically because you have a capital G represented, that dominant G, we would see colors. However, there's the capital F here, which is the allele, the gene that's going to prevent the production of colors. Since it's preventing the production of colors, we're not going to see colored chicken feathers, we're going to see white feathers. And in these first several examples, all of these have to have white chicken feathers because it has that capital F allele, which is the gene that is going to prevent the production of colors. So all of these are prevented from showing colors regardless of the color gene for the feathers. This one normally would show color, but it's being prevented. This one also would show color, but it's being prevented. This one would be white, but they're being prevented, so we're still seeing white. These last three, you'll notice that the F allele is recessive, so the production of color can come through. We can actually see the color. So now we have to take into account the gene allele. In this first one, we have homozygous dominant, so we're going to have the colored chicken feathers, and they're able to be expressed because it's a recessive F here. This next one, again, you have heterozygous for the colors of feathers, so we're going to be able to see colored feathers. They're able to be expressed because the preventing of the color is recessive, so those colors are able to come through. And then on the last one, again, you have the lowercase f allele indicating the colors can be expressed. However, the colors are homozygous recessive, and homozygous recessive is white feathers. So here you would see white feathers again. All right, a few more questions for you to answer if you'll pause the video and take a moment to answer those. Autosomal linkage The presence of two genes on the same autosome so that they tend to be inherited together and do not assort independently. The genes are linked. An example would be in fruit flies. The gene for body color, striped, and the gene for the antenna shape, having that feathery artistia. The body color, the capital E is going to give us a striped body. The recessive E is going to give us an ebony body. And then the antennas, the capital A is going to be a normal antenna, and the lowercase a for Aristopedia. Taking a look at our parent phenotypes, the male with striped body, normal antenna. These are going to be the genotypes that represent, and then the gamete. For the female, we have an ebony body with Aristopedia antenna. These are going to be the genotypes to represent, and then the gamete. So we would take our gametes and put the male on one side, female on the other. When they cross, you would get a striped body with a normal antenna. Now consider what would happen if a male from the F1 generation is crossed with a female. So this is from our F1 generation, our male, and then is crossed with a female with an ebony body and Aristopedia antenna. So our parent phenotypes are male, which was going to be striped body, normal antenna. That's what we obtained from here. And then the female had an ebony body with Aristopedia antenna. Again, separate out your gamete possibilities. You have two different possibilities here. So here are the male gametes and only one for the female. So there's the gamete choice for the female. And crossing them, we would get a striped body with a normal antenna and then an ebony body with Aristopedia antenna. All right, another question for you guys, if you'll pause the video and take a moment to answer that question. Autosomal linkage and crossing over. Offspring that show the same combinations of characteristics as their parents are called the parental type. Offspring that show different combinations of characteristics from their parents are called a recombinant. In this example, as you're looking at this, we have crossing over occurred in the female, so you're going to have a different combination, so you get the recombinant. You can see where this was the homologous chromosomes, crossing over has occurred, and so we can have that crossing over showing here. You can see by the capital letter, lowercase letter, as well as the mix of colors to kind of help you visualize that. In the male, crossing over did not occur, so the offspring combinations for as it's pulled down, it's going to be a parental type. It matches the same still. All right, the last questions for you guys, if you'll go ahead and make sure that you answer these.